Okay, so in the last video, we talked about glycolysis and converting that pyruvate into acetyl-CoA. So what we're going to be doing today is we're going to be finishing up this oxidative phosphorylation in citric acid or Krebs cycle. So if that pyruvate went into the mitochondria because there was oxygen present, these last two steps need to be taken care of. So we're going to look at what's occurring with both of those. So let's start first with the Krebs cycle. So the Krebs cycle, or the citric acid cycle, okay, so the Krebs cycle, citric acid cycle, this takes place in the mitochondrial matrix. Okay. So this is in the, oops, I can't spell. Basically, this is in kind of the liquidy type portion of the mitochondria. Okay, so this is taking place in the mitochondrial matrix. Okay, and what's happening during the Krebs cycle basically is that it's going to be releasing those leftover carbons from glucose. So those last couple carbons that were still there from the glucose molecule, as well as transferring energy to those electron carriers. And the carriers in this instance would be NAD, as well as FAD, and there was no FAD in our glycolysis process. So NAD, FAD, and ADP. There'll be a little bit of substrate level phosphorylation happening here, taking that ADP and converting it into ATP. Okay, so let's take an overview look here of what's happening during the Krebs cycle or citric acid cycle. You absolutely do not have to memorize all of these things. Don't even be writing this stuff down right now. Okay, we're just kind of overview, big picture. We're going to pick out a few parts of it, and then we're moving on. Okay, so it does start with the acetyl-CoA. There's that 2-carbon that the pyruvate acid was converted into when it lost the CO2. And you'll see there's transfer to the electron carriers. There's a little bit of substrate level phosphorylation happening here, okay, and it requires a little input of water. It's a metabolic process here, so it's going to require an input of water. Okay, so this whole cycle is happening here again in the mitochondrial matrix. And for every glucose molecule, it's going to turn 2 times for every glucose molecule. Because remember, 1 glucose gave me 2 pyruvates, which means I had to be given 2 acetyl-CoAs. So this cycle ends up turning 2 times per molecule of glucose. Okay, so let's get our basics down as what's happening during this Krebs cycle. So the first thing that we have happening is that the coenzyme A actually gets removed from the 2-carbon skeleton. And then that 2-carbon skeleton is actually attached. It's attached to an existing 4-carbon skeleton. So your acetyl-CoA, remember it had 2 carbons on it, and there's an existing 4-carbon skeleton there. So you actually end up with another 6-carbon chain. And of course that 6-carbon chain is going to be very quickly broken down. Okay? It's going to be very quickly broken down, and when it breaks down, it's going to release some CO2. Okay? Remember our equation for respiration? One of the products of respiration is CO2, and this is one of the places we get it. Okay, so the first thing we had here, okay, again, is the coenzyme A is released. That 2-carbon acetyl-CoA fragment there, that 2-carbon fragment attaches to an existing 4-carbon skeleton that's already there. These 6-carbon chains are now broken down. As they're broken down, they release carbon dioxide. And now we've got the electron carriers that come over. So now we've got some electron carriers. There's our NADH, our FADH2 coming over, and they're going to capture several electrons. So when they capture several electrons, remember that helps them harness energy, and they'll transfer that energy. Okay? And these electron carriers are going to be reduced, right? This NAD plus went to NADH, so it's positive charge was reduced. Same with the FAD. Okay, FAD went to the FADH2, so they were both reduced. Okay, now we've got the release of some more CO2 happening. Okay, so the citric acid or Krebs cycle releases quite a bit of CO2 here. And then as well as does a little bit of substrate level phosphorylation there for the formation of a couple of ATP. Okay. Basically, it produces 1 ATP per turn of the citric acid cycle. So per molecule of glucose, it's going to release 2 ATP molecules. Okay, so let's kind of summarize this again. Okay, so let's look at what we have here. So basically, this is kind of our energy summary of what's happening with the Krebs cycle. Okay, and these are the parts we care about. Remember on that picture I showed you a few minutes ago, it had all the different molecules and enzymes, and I told you that is not anything you have to memorize. Okay, so remember, each glucose molecule makes 2 acetyl-CoAs. Okay, and so this cycle turns 2 times because each molecule of acetyl-CoA turns that cycle. So when the acetyl-CoA comes in, remember the coenzyme A is removed, and these 2 carbon fragments are attached to an existing 4 carbon fragment. Okay, and so as that 6 carbon chain now gets broken down, I release CO2. Okay, the electron carriers come over and are able to start harnessing some of that energy, and CO2 is again also released, and we produce a little bit of ATP using that substrate level phosphorylation. Okay, so for every molecule of glucose, let's look at the numbers. Okay, so for 1 glucose molecule, I'm going to get out of the citric acid cycle, I'm going to get 6 NADHs. Okay, well, those are great energy transferring molecules and electron carriers, but so far I haven't really made a lot of usable energy yet, because I also got 2 FADH2s. Great, again, another electron carrier. Okay, I've got 2 ATPs out of the Krebs cycle. Okay, that's not very many, because right now so far I only have a total of 4 ATP. The 2 I got from glycolysis, and the 2 I'm getting here in the citric acid cycle. And then I get 4 molecules of CO2. Okay, so this is what my Krebs cycle will give me energy-wise, this up here. Okay, well, again, still not getting a whole bunch of usable energy yet. So that's where our electron transport chain is going to come into play. Okay, so our electron transport chain, along with chemiosmosis, okay, they make up the process here called oxidative phosphorylation. Okay, we've talked a little bit about substrate phosphorylation, substrate level phosphorylation, making a few ATP, taking those ADPs, giving them a phosphate, turning them into ATP. Oxidative phosphorylation will significantly increase those number of ATP, those usable energy molecules that are produced. Okay, so let's start with the electron transport chain portion of this. So those electron carriers that we've been talking about from glycolysis, some NADHs from glycolysis, the NADHs and FADH2s from the citric acid cycle, you can see them here, and they're starting at the beginning of the electron transport chain. So basically these electron carriers come over and they deliver their electrons to one of the first few proteins in the electron transport chain. You notice the NADH drops it off a little bit sooner than the FADH2. Okay, so the NADH actually will end up pumping even more hydrogen across the membrane than the FADH2 does. Okay, so these electrons are dropped off at the beginning of the electron transport chain, and then, like we talked about with photosynthesis, they are pulled along the electron transport chain until they reach the final electron acceptor, which in this case is oxygen. Okay, oxygen is the final electron acceptor. And remember, the electrons get pulled along the electron transport chain because as they move through the electron transport chain, they're moving toward molecules that are more and more electronegative. Okay, they have a stronger ability to pull electrons away from other molecules. And so that oxygen has the most electronegativity. So it's at the end of the chain, and it's helped pulling that electron transport, those electrons through that chain. That's how they move. Okay, this second protein here is this Q is more electronegative than the 1 or 2. Okay, cytochrome C is more electronegative than coenzyme Q. So as they move through the chain, they're moving from protein to protein that is more electronegative than the last one. And this is where we get our water. Remember, water is one of the byproducts of cellular respiration. So the CO2 is coming out of the Krebs cycle, or the citric acid cycle, and the water is coming out here during this oxidative phosphorylation as part of the electron transport chain. Okay, so let's look a little bit closer at the electron transport chain. So orienting yourself up here in the mitochondria, and you've got your outer mitochondrial membrane, the inner mitochondrial membrane. So there's the matrix there. Okay, and we'll look at a mitochondria again in just a minute so you can remember what it looks like with the folds of the membrane, increasing the surface area of that membrane, allowing for more of these electron transport chains. So you can see your NADH dropping off your electrons here. Okay, and as those electrons will move towards more and more electronegative molecules. So here's your electronegative oxygen here, accepting those electrons. Okay, and that will trigger the formation there of the water. Okay, as again, one of our products from this oxidative phosphorylation from cellular respiration. Okay, so now we need to look at how these electrons moving through the electron transport chain are actually going to create ATP. And that's going to be where that chemiosmosis is going to come into play. Okay, so let's focus now on this end chemiosmosis part, because that's where you'll notice you're actually making some ATP. So you'll notice as these electrons move through the electron transport chain, that causes hydrogen to be pumped across the inner mitochondrial membrane. And so what we're doing when that hydrogen gets pumped across the inner mitochondrial membrane is it makes a high concentration of hydrogen ions on one side of the membrane versus the other side. Basically, it creates a concentration gradient here. And when it creates a concentration gradient, those hydrogen then are naturally going to want to go back across the membrane to help even out that concentration gradient. And so the only way they can get back through is by going through that ATP synthase molecule. Again, just like in photosynthesis, the electrons move down the electron transport chain to create this hydrogen ion gradient. Those hydrogen ions then flowed back across the membrane through that ATP synthase, and as that ATP synthase spins, it is able to phosphorylate that ADP into ATP. And that's an example, again, of chemiosmosis. Okay, remember, chemiosmosis is when we use the energy of a hydrogen ion gradient to drive cellular work. Okay, so remember, that's our chemiosmosis. We're using the energy of that H plus gradient to be able to drive the work that's happening here, to be able to drive the cellular work. Okay, I'm sorry, my writing is so terrible today. So the energy that's stored in that hydrogen gradient across the membrane couples with those redox reactions of the electron transport chain, and this is how we're going to get our ATP synthesis here. Okay, it's essentially a proton, it's what's called a proton motive force. That's a V. Proton motive force. Okay, and so those hydrogen ions are responsible for establishing that. They create this proton motive force, which again is its capacity to be able to do work. And it's able to do work by flowing back through that ATP synthase. Let's take a little bit closer look at that. Both of these pictures show the same thing. We're just trying to get you to look at this ATP synthase. Okay, so you can see, again, that electron transport chain has helped create this hydrogen ion concentration gradient. So there is a large concentration of hydrogen ions in that intermembrane space. Okay, you see the matrix here where the Krebs cycle was taking place, citric acid cycle. So as these hydrogen ions now flow back through that ATP synthase, it'll spin around. Okay, you can see that rotor, it'll spin around. Okay, and as it spins around, basically that is what will attach those phosphates, those inorganic phosphates, to the ADP to create the ATP. This will generate about 10 million ATP per second. Okay, and if you look at your picture of a mitochondria again, you can see that the structure of the mitochondrial membrane is such that it has all these folds on the inside where you would have those electron transport chains and ATP synthase built in there. Out here is where I get my hydrogen ion concentration gradient. Okay, here's my matrix in here. Okay, and so by having those folds in that mitochondrial membrane, I'm increasing the surface of the mitochondrial membrane, allowing for more electron transport chains and ATP synthase. Okay, so never lose sight of your big picture. Remember, this is cellular respiration here, and this is respiration if oxygen is present. Okay, right? This is what will happen if there is oxygen available. If there is no oxygen available, then that's a totally different process. Right here, it becomes a different process. Okay, so we've got the glycolysis that produces the glucose turned into the pyruvate. The NADH picks up a few electrons. You see those NADHs are headed over here to the oxidative phosphorylation, to the electron transport chain and chemiosmosis. That pyruvate is turned into acetyl-CoA, which goes into the citric acid cycle, again releasing some more electrons, a little bit more ATP. Okay, so we got 2 here, 2 here. We're going to get 26 to 28 here. Okay, so we're going to get a significant number of them there. So oxidative phosphorylation is significantly better, a lot more efficient for actually producing lots and lots of ATP. And if we look at our glucose molecule, right, remember our glucose molecule was C6H12O6. Okay, so let's look at where those go real quick, right? Our carbons, well, they were released here in the form of CO2. Our hydrogens, they were transferred to the NADHs and the FADH2s. Okay, so that's where our hydrogens went. And our oxygens were also given off here in the CO2. Okay, so we've accounted also now for all of our atoms that were part of our glucose molecule that we started with. Okay, so again, this is cellular respiration when oxygen is present. Okay, and we'll be looking at fermentation next time.